be talking about microbead encapsulation of plant protoplasts as a tool for manipulating plant cell micromechanics. So the premise for our study was that currently biology lacks tools for manipulating the mechanics of individual cells. And this sort of tool would aid plant biology by allowing us to isolate mechanical variables at the cellular level, and that would allow us to start to determine how cells respond to their mechanical environments and how this might influence plant development. So we understand that mechanical signaling in plant development is important. Mechanical stress influencing microtubule orientation and cell wall microfibril deposition. And it also influences pin protein localization and auxin transport. These influence anisotropic growth, which changes tissue shape. And as tissue shape changes, mechanical stress in the tissue changes, and it feeds back into these. Unfortunately, mechanical stress at the tissue level is complex and difficult to understand. And so if we're to pick out an individual cell and try to understand what forces it's being subject to, it's influenced by both its own cell walls, its own geometry, and also the cell walls of the cells that are surrounding it. So one way to begin to tease apart these variables is to isolate plant protoplasts. We can do this by digesting the cell wall away, and it gives us naked membrane-bound cells. This is important because it releases the cells from the stress imposed by their own cell wall and the stresses imposed by the cells it's fused to. And it also makes isotropical spherical cells, and it standardizes the geometry of the system. So we propose droplet microfluidics as a way of fabricating these little microenvironments. So droplet microfluidics is a way of making small aqueous droplets by running a coarse stream of aqueous solution into two mineral oil streams that are coming from the top and the bottom. And this creates surface tension, and it breaks this coarse stream into microbeads or microdroplets of water. It's very high throughput. We can make a lot of droplets very quickly, and the droplets come out in a very consistent shape. So this is a video of this happening. We have agarose coming down here, oil comes in from the left and right, and the stream is broken into small droplets. These drop down into cooled oil and solidify, and we get solidified agarose microbeads. So this is our system, which is slightly more complex than the last picture. We have a commercial system manufactured by Dolomite, 3 pressure pumps drive fluid through microbore tubing that goes into our 2-reagent droplet chip. The oil comes in from the outside channels, cells come in from one of the central channels in aqueous solution, and agarose comes in, meeting the cells just before getting broken into droplets. It then drops down into a cooled oil bath where the beads solidify and contain protoplasts. So here are some pictures of the actual system. This is a dime for scale. This is our 2-reagent microdroplet chip. This is one of the pressure pumps. Another key to our system is this heating rig, which maintains our agarose at a liquid state and allows it to flow through these channels and into our chip before it's broken into droplets around here and then drops down into a cooled oil bath. Then we can separate these droplets out from the oil and put them into a culture medium. So here we are with our 2-reagent chip, cells coming in here, agarose coming down from up here, and we're capturing cells, and we're making about 130 droplets a second, so it's pretty good throughput. So as far as cell wall regeneration, these are our cells immediately after protoplasting and calcofluor staining, which you can't see because there's no cell wall. And then about 32 hours after protoplast isolation, around 10 hours after we capture them in microbeads, we can see a thin wall regenerate out over all the cells. So these captured protoplasts can then go on to divide, or they expand and grow and divide. So it's a suitable environment for them, and it allows us to look at how these mechanical environments are going to affect cell expansion and cell growth. After we do some fractionation by density, we can separate out most of the droplets that don't have cells and get around 50% capture percentage. So the ongoing research that we have to expand upon this is first to characterize the mechanical properties of our hydrogel materials using rheology. We also want to explore alternative materials and attempt to produce strong hydrogel matrices. And we're also looking to use different tissue. Everything that I just showed you is a suspension line of tobacco UI2 cells. They don't really develop beyond just dividing and dividing. And so some species with more developmental potential will be good to look into. So rheology allows us to quantitatively measure the mechanics of our hydrogels. So this is a rheometer performing a compression test on a small piece of agarose. And it's going to measure two things. It measures the force that's going through the agarose, and it measures the displacement. And so with that, we can calculate the elastic modulus of our material and begin to understand quantitatively what kind of environment we're exposing our cells to. Hydrogel engineering possibilities are also pretty diverse by virtue of drug release, consistent drug release, controlled drug release, I should say, and then tissue engineering. There's a lot of work going on in hydrogels. A couple of possible materials are methacrylated alginate and chitosan. So if we're looking at this as an agarose encapsulated cell, we could then capture this in something like alginate, methacrylated alginate, which can be cross-linked with calcium. Methacrylated alginate can also be cross-linked with green light. So we hit it with green light and it will cross-link further. And we can create a dynamic system that allows us to influence the mechanics of that system even after the cell is already captured. Alginate is also negatively charged, and so we can capture something around that like chitosan utilizing these ionic interactions and try to produce a strong chitosan shell. Chitosan is a derivative of chitin. Deacetylation of chitin leads to chitosan, which is then water soluble, and it can be used as a hydrogel. Chitosan can also be cross-linked covalently with something like genopin, and can also be further strengthened by raising the pH. So there's different stimuli responsive materials that we can use that will respond to different things. Another potential in these hydrogel engineering techniques is to use hydrogel blends. So chitosan gelatin blends have been explored for articular cartilage engineering. So very durable, strong materials. And one of the reasons that it's such a strong material is because not only does the gelatin solidify by temperature and can the chitosan be cross-linked, but the negatively charged gelatin will interact with positively charged chitosan and will create an even stronger matrix. We think that it's going to be important to have such a strong material because if we're really going to try to replicate the mechanical environment of a cell in vivo, plant cells can generate such enormous internal pressures. Cellulose is such a durable, strong material. And so that's sort of one of the tricks right now is how do we make a system that's so strong. Agarose being kind of soft, we really need a strong material to capture that cell in. So moving forward, we can begin to define the mechanics of these systems, like I said, quantitatively, and we can start to ask different questions about the force required for these cells to break out. What kind of force do they generate? If we understand the mechanics of this material well enough, maybe those are questions we can start to answer. Is it possible to surround a cell with such a strong matrix that we can impede it from further growth? And we can continue to take this in the developmental direction and see how developmental signaling at the cellular level is influenced by mechanical forces. So with that, I'd like to thank the Plant Biology Department of the University of Vermont, Phil Lintelak, my advisor, Gene Harris and Mary Tierney on my committee, Rachel Oldinsky, who's in the College of Engineering and Math Sciences at the University of Vermont, who is doing a lot of work with the hydrogel engineering side of things, and Patrick Sharon is her lab tech. If you're interested in this, we have a recent publication in Applications in Plant Sciences where we essentially tell you how you can do this yourself. So that's all I have. Thanks.